There, I think we're ready now. Good morning, everyone. If you are joining us today on the video, there are a few videos we're going to show on YouTube that I'm going to point will be up here on the screen. Realize those are also listed today in the in-person agenda. So we have a test next week, Thursday on the 5th, which is also the last day of the quarter. So this test will go on this quarter's quarter grade. Be chipping away at all your Canvas things along the way. I want to kind of refresh your memory on an activity you did. I think it was last week, Thursday or Friday. These are one of those POGIL activities. And we talked about different types of movement of molecules. So there's this idea called simple diffusion. And simple diffusion is just diffusion. So these molecules are going to go from high concentration to low concentration across a semi-permeable membrane, okay, or through a membrane. So the analogy, I don't know how long. That's the cord I get. No, it's like all the cord I really do get. All right, so analogy that I kind of like to use is this idea of a mosh pit. You guys familiar with what a mosh pit is? Okay, you probably have been to a Mozart concert or Beethoven and gone crazy in a mosh pit. Now, obviously, I know it'd be a cover band because Mozart and Beethoven are dead. I know. So, cover band for them. But the idea is when you're at a concert, sometimes people go a little nuts. They get really into the music and they're like, whoa, and then they get in this mosh pit. And a mosh pit, if you aren't familiar with what a mosh pit is this kind of crazy, random pushing, shoving, bouncing off of each other. That's kind of a nice way to describe it. It's
pretty
chaos.
intense.
Huh?
Chaos.
Chaos, yeah.
It is chaos.
It is chaotic. And there's not a lot of rhyme or reason to where you, if you were ever in one, you don't have a whole lot of control about where you are going, right? You're kind of getting shoved, you're shoving people, bouncing off of each other, okay? So let's imagine for a minute that you guys are in a mosh pit in the hallway because you're so excited to get into biology. Right. What are you guys laughing at? It's a thing.
I don't think we have to imagine that. I think that's reality.
Yeah. I don't know why they were laughing at that. I mean, I've seen it. You guys were, good thing the door was open when you came, otherwise probably would have happened today, right? So let's pretend that you guys are outside and we put a few more walls up, right? So maybe we put a wall like right here in the hallway and then maybe like right here so we can confine you a little bit. Now why? Because when we confine you, you're gonna hit the walls more often. There's gonna be more collisions, okay? So you guys are out here going like, whoa, mosh pit, can't wait for bio. But the door is closed, okay? Can you come in? Mm-hmm. Can anything come in this room?
Mm-hmm. Like air.
Air, so some things can come through, right? So this door, we could almost say is a semi-permeable membrane, right?
Oh, you gifted them.
Oh, you're right. Well, so some things can go through, but others things can't. What would we call it?
Semi-permeable.
Membrane, right? Or selectively, right? Those are kind of interchangeable. Okay?
My bad.
What?
I said my bad.
No, you're perfectly right. No, semi-permeable and selectively permeable, I'm saying, are like one and the same. They're just another way to say the same.
Okay.
Okay, so you guys are, bio! And I'm like, that's in the camera. I don't know if they can actually see me, but, and then I go like, I open the door. So what happens? You guys are like, woo, bio! And some of you like are going like, I'm going to bio! Nope, mixed it. Right? Because you hit the wall. And you don't have a whole lot of control over what you do. But some of you lucky folks are going to be like, hey, I'm going in the right direction. I made it to bio! Right? But you don't have a whole lot of control over what you did. Constant random motion. Mosh pit. Compared to people, you guys out here, and me and Mr. DeBoer inside, just like, I know they're eager to learn. I'm a little concerned about opening this door. What's true about concentration of people? Where is it high? Or where is it higher? Outside the room or inside the room?
Outside.
Outside the room. Okay? Now when I open that door, which way are people going to flow?
This way.
Into the room, right? From high concentration to low concentration. Is that easy or hard to do if you're mosh pitting out there to fall into the room? Does it take a lot of energy?
No.
No, right? Because it's this, okay? That's diffusion. That's diffusion. High concentration to low concentration. Hi, Barb. I almost whacked Barb in the face. Yep. Yep. Not until that time goes. So, high concentration doesn't take a lot of energy. That's diffusion. Now, there's two types of diffusion. What this called simple diffusion is what water, sugar, salt do. They just go through the phospholipid bilayer. Okay, goes through the phospholipid bilayer. The second page of that, Mr. DeBoer, I think has the diagram right there. Okay, so that's a diagram that you should have a pretty good, I'm gonna say a basic, let me rephrase it. You should have a general knowledge of, is a diagram like that. One of the things we asked you to do on this worksheet was to draw this, and it says, note, you will need to be able to do this for your test. Okay, so this is a very complicated structure. Okay, but the basic idea, now they call them slightly different things than what we will in our other notes are, but we have these things right here, and if we look at our key, hey look, that's a phospholipid. And we say, hey, look, there's a phospholipid and a phospholipid, and there's two layers of them, so we call it a phospholipid bilayer. Mr. DeBoer is going to draw a little bit more simplified picture of what we are really concerned about. And then they have these things right here. Now they call them membrane-spanning proteins. I don't call them membrane-spanning proteins. What I tend to call them is transport proteins, or a protein channel is more commonly what I call them, is a protein channel. Okay. Now, if we are talking about the door is kind of a transport protein or a protein channel. And in a situation where air could fill underneath the gaps, they're going straight through that phospholipid bilayer. But you are too big to go through that phospholipid bilayer. What you need is something called, and this is on the next page of this that you already did, because this was your homework last week. You need some help, okay, to get through the door. And we need these things called like protein channels or transport proteins or membrane-spanning proteins, but that's not really what we ever really call them besides in that one worksheet. But the idea is if you are in an argument with someone, let's say it's a contract negotiation, you're trying to figure out how much they should pay you. If you and your boss can't settle and decide upon how much to pay you, they might bring in a facilitator or an arbitrator. And the idea is they're going to aid you or help you with your dispute. So when we talk about facilitated diffusion, it's still high concentration to low concentration, mosh pit outside to non-mosh pit inside. The idea is that with facilitated diffusion, you need some help. And that's all it is. It's still high to low concentration, doesn't take a lot of energy. You open the door, whoa, you fall in, okay? There's other things involved here, but that's the basic idea. Okay, so that is what we generally refer to as simple diffusion or facilitated diffusion. Now, it was easy. It didn't take a lot of energy from you to go from high energy up here to low energy, concentration. High concentration to low concentration. It took very little energy. I like to drink coffee in the morning. And sometimes, and this is maybe a little bit more personal information than you need, but sometimes I have to go to the bathroom after I drink coffee. It's part of life, right? So class is about to start. You guys are like, biology, mosh pit out there. And I'm like, I really gotta go to the bathroom. I really gotta go to the bathroom. I gotta go to the bathroom before class. So I can be like, all right, you guys can go to class. I gotta go to the bathroom. How easy is it for me to go out the door when you guys are mosh pitting out there?
Not very easy.
Not very easy, right? So there's terms that we use like with the concentration gradient or down the concentration gradient. And there's a couple different analogies. We talk about like a slide. Because you have a high energy, you're at the top of the slide, down here is low, you're going down the slide. The bigger the concentration, the more people, the differences in concentration, the steeper that slide is. You can also, so it's easy to go down the slide, but it's hard to go up that slide or against the concentration gradient. You could use a river analogy. The river flows from upstream, downstream, right? That's easy, it doesn't take a lot of energy. Trying to paddle my canoe or swim upstream takes a lot of energy. So for me to go against that concentration gradient, from low concentration to high concentration, that's hard. It takes a lot of energy, which is what we say for active transport. If I were to describe my children, and I didn't want to be like really, really rude, I'd say my children are very active in the afternoon. And what I'm really saying is they're a little crazy and they have a lot of energy, right? It's just a more polite way to say. You don't say like your kid's like nuts. I don't mean in the standpoint of like mental health. I'm saying like energy-wise, like whoa, they are going nuts. Like they have a lot of energy. They're very active, is what we politely say to other parents. That means energy. Okay, they have a lot of energy. So to go from low concentration to high concentration is called active transport, and it requires a transport protein or a protein channel to go across the cell membrane. Okay, so those are just a couple kind of things that we are going to, oops, I forgot to put those up. So the sheet, some of you I do notice grabbed the sheet on the back. However, I would have assumed you probably would have noticed that it's not actually a biology sheet. This is actually a physics sheet based on the fact that it says 2D motion. So if you grab one of those, you can actually grab the wrong sheet. Okay, so later in class, you can grab that section 4. Section 4, guided notes. It's our last section. It's your homework. Mr. DeGrand, another biology teacher, recorded a video for you to kind of walk you through that. Okay, you can get that a little bit later. But those ideas are talking about how do we continue to move particles, some of which you've already talked about. And you've already done some learning about with these active transport stuff. Okay, so this is also from, I think this is section 3 notes. This picture is not nearly as nice as Mr. DeBoer's, but it's the same, has some of the same things. Now there is a few things on here. They have these carbohydrate chains, and these carbohydrate chains are something we're going to talk about a little bit today. And they're used for like some cell-to-cell signaling in between cells or viruses entering the cell. Okay, so we're kind of going to use that as our transition. There's 2 types of transportation of big molecules in cells that section 4 guided notes will talk about. There's something called endocytosis, which endo is when things go into the cell. Endo, into. Then there's something called exocytosis, and exocytosis is where these big molecules exit. Exocytosis, exit the cell. Okay, and we'll kind of use those terms a little bit in our discussion today. So, coronavirus, SARS-CoV-2. This is what a general structure of a SARS-CoV-2 or coronavirus looks like. And if we look at these, there's a few things that we'll kind of notice, and we'll kind of ignore some other things. But if we look at these red molecules that are kind of going around, you can say, boy, there's like this circle on each side, and then there's these pegs. And what all they call it in this diagram is they call it a membrane. But what these membranes are is they're a phospholipid bilayer. SARS-CoV-2 is surrounded by a phospholipid bilayer. So what happens when SARS-CoV-2, it merges with your phospholipid bilayer for endocytosis. Okay, now it doesn't just enter immediately. There's those spike proteins that they often talk about. This is kind of the classic picture of SARS-CoV-2 that we often see is that. And those red things are what this is referring to as surface protein. Okay, these things right here are the same things that we saw there. They're proteins, or they're spike glycoproteins. Okay, now what happens? Basic structure, basic infection. Oops. How do your cells get SARS-CoV-2 inside them? So this is what that structure that I just showed you was. This is our phospholipid bilayer. Notice how they put 2 layers because there's a bilayer there. We have these things called ACE2s. The idea is they're those carbohydrate chains. It's a point where the virus can attach to the cell membrane, and it's like, hey, this is a key to a lock. And I've got a video that I'll reference, that I'll show you that I'll kind of reference. Then once that happens, this kind of gets pulled in, and then it merges with the cell. It goes into endocytosis, and then I'm just going to very crudely say what happens when it opens up. It spews its contents, it goes into the cell. Now what stuff? I kind of forgot that. This stuff. And what this is genetic information. It's RNA. Now what we have is we have DNA. DNA makes RNA. RNA then goes to your ribosomes. We'll come back to that in 1 second. I'm going to ask, what does a ribosome do in about 20 seconds? So what happens is this virus enters the cell membrane, it spews its genetic RNA information into the cell, and it goes to the ribosome. What is the function? You should know this. What is the function of a ribosome? It makes proteins. It makes proteins. Now it makes your proteins. But what happens is this RNA comes to your ribosomes and they hijack your ribosomes and it starts making their proteins. Okay, it starts making the virus's proteins. And then those go to your endoplasmic reticulum. They get packaged for the shipping department in the Golgi apparatus. And then what happens is it forms something called a vesicle, and it goes to your cell membrane and it exocytosis and it spews the content back out. Endo into, exo exit. Okay? And you can say, boy, that sure does look a lot like that, doesn't it? And he's like, so what? One virus comes in, one virus goes out. Big deal. No. One virus goes in, lots of viruses come out. And then those viruses, one virus goes into a cell and produces lots of viruses, which are released to go to the next cells. And then it's exponential growth. You get infected with said virus. Okay? That's kind of the big, but it hijacks our cells. It uses our cells' organelles to do what our cells are designed to, but it steals them, it hijacks them. So this video, if I can pause it at the right time. Oh, I missed it.
I'll take a couple shots at her. Hey, you're not gonna become an epidemiologist after watching this, but the 2 videos actually do a really nice job of explaining what happens if you would get COVID and why can.
Not all
Yes,
people.
good point.
Symptoms are very different with COVID, right? We've heard this. Some people are what they call asymptomatic. They get a fever and a headache for like a day, and then like I feel perfectly fine, but they still have COVID. And there's other people that are very different than that. I mean, hospital-ridden, they're on a ventilator, they die. Okay, so very different. So what could happen, right?
But it's good info, and it's not at some doctorate PhD level where like you can watch and go, oh, okay, I understand that. I have a basic understanding of some of this biology, and as a result, make some sense of what's happening.
Achoo!
Nice job.
Thank you.
I think it was even better than yesterday's.
Achoo! I always look at that and it's kind of gross. You know, it's gross. Unfortunately, we've all been too close to our friends at some point in our lives where that happens and you get sprayed and you're like, ah, right? And now, we've always thought that's gross, but now I think very differently about a picture like that than I would have like 1 year ago today. Okay? But what happens is you sneeze, these tiny droplets get very violently expelled from your body. Now this is a sneeze, same thing happens with a cough, same thing happens when we talk. when we sing, okay, things like when we breathe, absolutely. So I kind of jokingly in the past have talked about people in the front row kind of being in the spit zone or the splash zone, because we've all unfortunately had the situation where we're too close to someone, all of a sudden this little tiny gob of spit comes flying out of our mouths and lands like on their mouths, right? So that happens sometimes, right? And we can visibly see that. The thing is, that's a big droplet we get some. Small droplets happen all the time, even when we aren't like big gob. This stuff is coming out of our mouths, okay? So why are we wearing cloth masks? And there's some videos out there, not super scientific, but they visually kind of do a nice job. They use like a can of spray paint and they put it behind a mask. What's the purpose of our cloth masks? Yeah.
They catch the water.
Absolutely, right? If we were to take, and more than droplets, let's just call them droplets, because water droplets, virus droplets, all things included. If we were to put a mask, say like right here, this should have gotten trapped on here. Okay? Those particles should get trapped in our cloth masks. And in the event that you have a virus, you have the virus, the COVID or SARS-CoV-2 virus, they should get trapped in your mask. We wear masks not as a filter. These masks, let me rephrase it. The function of these cloth masks is not to, there is some filtering, but the main benefit is not to filter your air coming in. It's to filter your air coming out. It's to prevent these from getting to your neighbor or your friend. Okay? So that's why we wear a mask. Now what happens once these things get expelled? Gravity takes over and then they go, phew. Why are your desks as far away as they are? Because statistically, those droplets will have hit the ground before they get to you. That's why we get 6 feet. That's why they say 3 to 6, right? Is because in theory, that little droplet should have gone like, even if I go, like even if I was really good at spitting, and I don't know if I could spit as far as Mr. DeBoer is away, right? Because gravity's gonna, and that'd be gross. I wouldn't actually spit at Mr. DeBoer. Okay, that'd be not, but that's the idea. Okay, not today. Last year maybe I would have, but not this year. Okay, I'll try to be quiet for a little bit. I find it hard.
This is SARS-CoV-2. It belongs to the family of coronaviruses named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. What actually happens in your body when you contract the coronavirus? What exactly causes your body to develop pneumonia? And how would a vaccine work? The coronavirus must infect living cells in order to reproduce. Let's have a closer look. Inside the virus, genetic material contains the information to make more copies of itself. A protein shell provides a hard protective enclosure for the genetic material as the virus travels between the people it infects. An outer envelope allows the virus to infect cells by merging with the cell's outer membrane. Projecting from the envelope are spikes of protein molecules. Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery, repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action...
Key in the lock, hey, I'm the right person, let me in. Okay, and now what's happening is this is endocytosis. The virus is going into the cell, and once it gets inside, it's gonna like spew its contents, and it's gonna release them.
Action allows the virus to get inside your cell. A typical flu virus would travel inside a sac made from your cell membrane to your cell's nucleus that houses all its genetic material. The coronavirus, on the other hand, doesn't need to enter the host cell nucleus. It can directly access parts of the host cell called ribosomes.
Which does what? Make proteins.
Ribosomes use genetic information from the virus to make viral proteins, such as the spikes on the virus's surface. A packaging structure in your cell then
The packaging structure in your cell that gets things ready for export is called, like the shipping department, is called the...
the spikes in
Oh,
vesicles
Golgi
which
apparatus.
merge with your cell's outer layer, the cell membrane. All the parts needed to create a new virus gather just beneath your cell's membrane. Then,
Exocytosis.
a new virus begins to bud off from the cell's membrane. Now, with the virus spreading in your body, how can you develop pneumonia symptoms? For this, we'll have to look into your lungs. Each lung has separate sections called lobes. Normally, as you breathe, air moves freely through your trachea or windpipe, then through large tubes called bronchi, through smaller tubes called bronchioles, and finally into tiny sacs called alveoli. Your airways and alveoli are flexible and springy. When you breathe in, each air sac inflates like a small balloon. And when you exhale, the sacs deflate. Small blood vessels called capillaries surround your alveoli. Oxygen from the air you breathe passes into your capillaries, and then carbon dioxide from your body.
Diffusion, simple diffusion, high concentration to low concentration. Now, if we were talking specifically about water, the diffusion of water is called osmosis. It's still diffusion, it's still high concentration, low concentration, it's just very specific to water.
It passes out of your capillaries into your alveoli so that your lungs can get rid of it when you exhale. Your airways catch most germs in the mucus that lines your trachea, bronchi, and bronchioles. In a healthy body, hair-like cilia lining the tubes constantly push the mucus and germs out of your airways, where you might expel them by coughing. Normally, cells of your immune system attack viruses and germs that make it past your mucus and cilia and enter your alveoli. However, if your immune system is weakened, like in the case of a coronavirus infection, the virus can overwhelm your immune cells, and your bronchioles and alveoli become inflamed as your immune system attacks the multiplying viruses. The inflammation can cause your alveoli to fill with fluid, making it difficult for your body to get the oxygen.
What do we call fluid in your lungs? Just very generic term, I think they've already mentioned it,
There's the P.
but it doesn't sound like it starts
What?
pneumonia, right? So when you have fluid on your lungs or fluid in your lungs, they often call, hey, you have pneumonia. We get pneumonia from lots of things. It doesn't have to be from coronavirus, but that's one of the things we can get.
I was gonna say phlegm, but that's like in your throat.
So when they actually talked about like those cilia will push the mucus out, that's exactly what it is. They were talking about phlegm. So every day you have phlegm that gets expelled out of your lungs, and there's these little cilia, and a lot of times we don't really notice it because it just kind of pushes them up your bronchioles, and once it kind of get your esophagus and your bronchioles are kind of merged, there's a valve, like, but they, air and food go down your throat, and then they split. So a lot of times those cilia just kind of push it back past that part, and then you swallow it without you actually knowing most of the time. Now sometimes we get it up to our throat, and that's when we like cough up phlegm or stuff like that, and when we're sick there's more of it. But that's exactly what we're talking about, is phlegm and mucus is what they were talking about. But this is just fluid buildup in your lungs. It could be viral caused, it could be bacterial caused. There's lots of different reasons why you can get pneumonia.
Oxygen it needs. You could develop lobar pneumonia, where one lobe of your lungs is affected, or you could have bronchopneumonia that affects many areas of both lungs. Pneumonia may cause difficulty breathing, chest pain, coughing, fever and chills, confusion, headache, muscle pain, and fatigue. It can also lead to more serious complications. Respiratory failure occurs when your breathing becomes so difficult that you need a machine called a ventilator to help you breathe. These are the...
Why do you keep pausing the video, Mr. Davies? Just because I want to try to make some point. So back in March, April, we heard in the news, we used to hear a fair amount about ventilators, I feel like at least. And they would talk about how people are getting sick and they have to get put on a vent or they have to get put on a ventilator. And then basically what it does is this breathes for you. So the previous thing showed a tube going down, it's sealed off, and basically it pumps air into you, it forces air into your lungs to cause them to expand, and then it pulls air out, which causes them to contract. Because your breathing is labored, you're like, it's hard for you to do that. So the idea is if we take a machine, we force air in and force air out, it can breathe kind of for you. Early in like the pandemic, we heard stories about like New York City was getting really hard hit. Their their hospitals were getting full and overrun, and they didn't have enough ventilators for the patients that needed them. So then there was like debates about like, well, upstate New York had some and they weren't getting very hard hit. Should they be able to go take those from those hospitals because they aren't using them yet? And that's why they're kind of the news. They've ramped up some production, or they were going to ramp up some production of ventilators. I don't hear a lot about ventilators anymore. So one thing I did actually hear about ventilators was today in the news, part of the whole Foxconn thing is there was talk about whether Foxconn was actually going to transition and team up with a company called Medline to start producing ventilators. Whether they did or not, that was kind of what the story was about. But there was companies transitioning to try to help with ventilators because that's one of our treatment of symptoms with COVID.
Are the machines that save lives and that medical device companies currently ramp up production for. Whether you would develop these symptoms depends on a lot of factors, such as your age and whether you already have an existing condition. While this all sounds scary, the push to develop a coronavirus vaccine is moving at high speed. Studies of other coronaviruses led most researchers to assume that people who have recovered from a SARS-CoV-2 infection could be protected from reinfection for a period of time. But that assumption needs to be backed by empirical evidence, and some studies suggest otherwise. There are several different approaches for a potential vaccine against the coronavirus. The basic idea is that you would get a shot that contains faint versions of the virus. The vaccine would expose your body to a version of the virus that is too weak to cause infection, but just strong enough to stimulate an immune response. Within a few weeks, cells in your immune system would make markers called antibodies, which would be specific for only the coronavirus, or specifically its spike protein. Antibodies then attach to the virus and prevent it from attaching to your cells.
Okay, so those little antibodies block those, or attach to the spikes, and those spikes were the key to get into our cell. But if we put little spikes on them, now our cell doesn't recognize them and our cell doesn't let them in. That's the idea behind that.
The immune system then responds to signals from the antibodies by consuming and destroying the clumps of viruses. If you then catch the real virus at a later stage, your body would recognize it and destroy it. In other words, your immune system is now primed. Collecting evidence on whether this will be possible, safe, and effective is part of what's taking researchers so long to develop a vaccine. It's a race against time to develop a vaccine amid a pandemic. Each step in vaccine development usually takes months, if not years. An Ebola vaccine broke records by being ready in 5 years. The hope here is to develop one for the new coronavirus in a record-breaking 12 to 18 months. While all of this will take time, stay home if you can to protect the most vulnerable. And don't forget to wash your hands for at least 20 seconds and as often as possible. This video was a collaboration between Nucleus Medical
So what do you think? Washing your hands.
Media and the What If channel, where we usually dive into.
There we go.
20 seconds, is 10 good enough? 15 good enough? Think so? Yeah? Well, let's take a look.
Best way to prevent the spread of coronavirus is to wash your hands.
Wash your hands.
Wash your hands! But why? It's because soap, Regular soap, fancy honeysuckle soap, artisan peppermint soap, just any soap absolutely annihilates viruses like the coronavirus. Here's how. This is what a virus, like coronavirus, looks like. It's a bit of material surrounded by a coating of proteins and fat. Viruses easily stick to places like your hands, but when you rinse your hands with just water, it rushes right over the virus. That's because that layer of fat makes the virus behave kind of like a drop of oil. You can see it happening in this demonstration. Oils are just liquid fats. What happens when you pour oil into water? It floats. It doesn't mix. But add soap, and suddenly that fatty oil dissolves into the water. That's because inside, soap has two-sided molecules. One end of the molecule is attracted to water, the other end to fat. So when the soap molecules come in contact with water and fat, these dual attractions literally pull the fat apart, surrounding the oil particles and dispersing them through the water. Let's go back to our coronavirus molecule with that layer of fat holding everything together. When it interacts with soap, bam, that fat gets pulled out by this.
So if we start pulling apart the membrane of the coronavirus, we're pulling the spikes away, and those spikes are what interacts with their cell membranes, which then inserts its genetic information into you. But if you start pulling that apart, now it's like a water balloon, that if you pull a chunk of the water balloon out, how well does that water balloon hold water? Well, it doesn't hold it, right? So how well does it keep the genetic information? Then it wants to spew it, because it's got to get into your cells. And if this is the water balloon and we start pulling chunks, we're going to start squirting water all over the place, and they aren't going to have a mechanism to get into our cell.
The soap. Soap literally pulls apart and demolishes these viruses. And then the water rinses the harmless leftover shards of virus down the drain. But, and you know where I'm going with this, it takes time for this effect to happen. 20 seconds to be specific.
Morgan.
To show why, we ordered this lotion that mimics viruses and their fatty layers. It glows under a UV light. If you just rinse your hands under regular water, nothing comes off. If you wash with soap for just 5 seconds or 10 seconds, your hands are still covered. The virus is still there, able to get you and others sick. But 20 full seconds, now the soap is actually destroying the virus. Hand sanitizer works too because it's mostly alcohol, and alcohol works in a somewhat similar way to soap, breaking down that fatty layer. You need a high concentration of alcohol to make that work. The CDC recommends hand sanitizers with at least 60% alcohol. But even with 60% alcohol, the CDC recommends using soap if you can. If your hands are sweaty or dirty when you use the sanitizer, that can dilute it and diminish its effectiveness. As for soap, just any old soap works. You don't need soap marketed as antibacterial even. The FDA says skip it. There's no proof it's any more effective. Just be sure to wash your hands for 20 seconds. That's happy birthday twice.
Happy birthday, dear. I guess me.
Or the chorus to Lizzo's Truth Hurts.
Straight to my face.
Or Prince.
Raspberry beret.
Or Eminem.
Go, go, only go.
Or even Dolly.
Jolie, Jolie, Jolie.
Just as long as it's 20 seconds. And you're using the ultimate virus annihilator. Soap.
Can you turn the lights off?
I can.
May you?
I may.
Would you please?
No, but thanks for asking.
So I was trying to look. I don't see it quick popping up.
What's the quick and easy way for you as an individual to help increase the likelihood of not catching that virus, or any virus for that matter? Because that hand wash, that's not exclusive to coronavirus, okay? Like, that's not, hey, well, if it's the 20-second hand wash with soap, then I won't get corona. right? That could be a lot of other viruses, and there's plenty out there. But thank you for playing along, Morgan, when I said, does it have to be 20 seconds? Is 10 seconds good enough? Is 15 seconds? And she's like, yep, 10, 15 seconds good enough.
Yeah.
At least 20 seconds. Gotta give the soap time to work, do its job to break down that outer layer. 60.
Shoot, I forgot. 62% alcohol in Purell is according to this random website that I looked. So Purell, I'm saying Purell because that's what we have in front of our room, is 62%. And they say you should have at least 60%. Okay? So the idea is we can break down that membrane. If we break down the membrane, we just took apart the part that allows the genetic information to enter our bodies. Okay? So, yeah, we have about 10 minutes, or I guess questions. Okay, so there's kind of twofold, a little bit of a coronavirus kind of background, but also how it interacts with, how does it get into our cell, endocytosis, how does it leave, exocytosis? What happens once it gets in? It uses our cells' organelles to produce proteins, okay? So with that, we have about 10 minutes left. There's stuff that you may not have done yet, okay? You may have some late work, and now would be a great opportunity to say like, hey, I didn't really understand this. Or there's that osmosis lab with the potatoes. I didn't really understand the questions of those. Great opportunity for you to get some help on those. We can get those things done. We can get those things submitted, and we can move on and get ready for our test next week, Thursday. If you're all done with that stuff, on that back table is section 4 guided notes.
Yeah, don't forget to grab that.
Okay, if you want to do the paper copy of section 4 guided notes, then make sure you grab a paper copy from that back table. It is linked as a Google Doc, it is linked as a PDF as well, but those are things that you should be working on. So either late work or get a start on tomorrow's Mondays, because you don't have school tomorrow, okay? So Monday is your one virtual day for the week of this week, whatever this week is, okay? So that is your virtual day assignment. There's one day of virtual assignment, and that is your virtual day. Get your missing stuff taken care of. That is gonna help prepare you for your test. That is next week, Wednesday or Thursday, depending if you're A-Day or B-Day. I was looking at the camera for that. As for you A-Day kids in virtual land, if you have questions, please let us know. And cut.